Hello everyone, welcome to the Zebrafish Genetics Laboratory, Department of Genetic Engineering, SRM Institute of Science and Technology. I am Kiran Kumar, an Associate Professor in the Department of Genetic Engineering, and today I am going to present to you a demonstration video on the protocol for anesthetization of adult zebrafish. Anesthesia is a reversible state of controlled sedation which is used to minimize pain and stress. Laboratory animal models are anesthetized to ensure humane handling for imaging or minor surgical procedures. Zebrafish, also known as Daniel Herrero, is widely used as a vertebrate model system as they share a high degree of genetic and physiological similarity with humans. Adult zebrafish are commonly anesthetized using immersion technique via two methods. The first one being chemical method, the second one being physical method. Chemical method. The generally used anesthetics for adult zebrafish include drugs like MS-222, eugenol, benzocaine, and isoflurane. MS-222, also known as Tricaine methane sulfonate, is the most common chemical anesthetic used in zebrafish research, and it acts via blocking voltage-gated sodium channels. MS-222 is hypothesized to block the neural signaling to the brain. MS-222 is water-soluble and hence easily administered in aquatic environments, but requires buffering with sodium bicarbonate to maintain pH stability. Preparation of anesthetic solution for MS-222. MS-222 is a derivative of benzocaine with an additional sulfonate radical, making it more water-soluble and more acidic than its parent compound. MS-222 is observed to have no effect on ciliary axon, but its effect on muscular activity has been observed to be rapid. To prepare 0.5 millimolar of tricaine stock solution, 81.75 milligram of MS-222 is dissolved in 1 liter of distilled water. The pH of the solution needs to be adjusted to 7 to 7.5 to avoid acidity-related stress for the zebrafish. Anesthetization procedure with MS-222 involves three major steps: immersion and induction with the chemical anesthetic, monitoring and maintenance of the experimental zebrafish, recovery and post-anesthesia care for the experimental zebrafish. Let us look at the first step, immersion and induction. The experimental zebrafish need to be exposed to MS-222 bath. The working concentration of MS-222 for adult zebrafish is 0.01 millimolar. Induction usually occurs within 30 seconds to 2 minutes. Signs of proper anesthesia are decreased opercular movement, loss of writing reflex, reduced response to stimuli. Now let us look at the parameters for monitoring and maintenance. We need to continuously observe the experimental fish to ensure proper depth of anesthesia. If anesthesia needs to be prolonged, fresh oxygenated anesthetic solution might be required. Now let us go on to the final step, which is the recovery and post-anesthesia care. Zebrafish are transferred to fresh aerated system water. The recovery for the experimental zebrafish is typically very rapid within minutes. Full recovery is confirmed by normal swimming behavior and proper response to any external stimuli. Depending on dose and duration of exposure to MS-222, the zebrafish anesthesia involves 4 stages. First stage, light sedation. Experimental zebrafish becomes less active but retains body balance. The animal is still responsive to stimuli. Second stage, anesthesia. It is a deeper level of unconsciousness where the experimental zebrafish is no longer responsive to stimuli. Third stage, surgical anesthesia. The experimental fish will be in deep state of unconsciousness, and this stage is suitable for minor surgical procedures. Fourth stage, overdose. If the exposure is high, the test zebrafish can enter a state of overdose which may lead to death. The opercular movements stop for the experimental fish in this stage. Physical method. Hypothermic anesthesia or cold induced sedation is a non chemical method of anesthetizing zebrafish by lowering water temperature. Rapid exposure to chilled water, 4 to 10 degrees Celsius, induces a state of sedation by slowing down both metabolic and neurological activity. This technique is ideal for short, non invasive procedures that require quick immobilization. The limitation of this approach is the potential for a stress response as well as the variability in its effectiveness across different individuals. Anesthetization procedure using hypothermia involves 4 major steps: preparation of cold water bath, induction, monitoring during anesthesia, recovery process. Preparation of cold water bath. System water is chilled to 4 to 10 degrees Celsius using ice or a cooling system. A separate recovery tank with standard system water with a temperature of 25 to 28 degrees Celsius should be prepared and maintained. Induction. Adult zebrafish are transferred into the chilled water. Sedation typically occurs within 1 to 3 minutes with fish displaying reduced swimming activity. Monitoring during anesthesia. Opercular movement is continuously observed. Recovery process. Zebrafish are immediately transferred to a pre-warmed aerated recovery tank. Recovery usually occurs within 1 to 5 minutes with fish regaining normal swimming behavior. In conclusion, anesthesia in adult zebrafish is a crucial aspect of experimental procedures enabling humane handling while minimizing physiological disturbances. Tricaine is the gold standard for zebrafish anesthesia due to its effectiveness, reversibility, and widespread use. Physical anesthetization serves as a quick alternative for brief procedures. The choice of method should be based on the procedure type, ethical considerations to ensure minimal stress and humane treatment of the experimental zebrafish. Thank you for watching.